Welcome to Learning Science is Fun. I am Wayne and today we will learn about angiotensinogen. In this video, we will discuss what is angiotensinogen, what is the function and mechanism of angiotensinogen, and what happens when angiotensinogen is impaired. Without further ado, let's get started. What is angiotensinogen? Angiotensinogen is a 485 amino acid with a molecular weight of 1,759 grams per mole. It is also known by the names renin substrate, proangiotensinogen, and serpin clade A member 8 or serpin A8. Angiotensinogen belongs to the serpin superfamily of proteins, known for its protease inhibiting effects. It is also the sole precursor of all angiotensin peptides. The crystal structure of glycosylated angiotensinogen is shown in Figure A. It shows different protein folding including helixes and alpha and beta sheets. It is known that angiotensinogen is a sole precursor of several angiotensin peptides, including angiotensin 1 and angiotensin 2. Angiotensin 1, which is colored purple in the figure, is a decapeptide, forming the first 10 amino acid of the angiotensinogen protein from asparagine 1 to leucine 10. It is later cleaved by renin between leucine 10 and valine 11 to release it. Little is known in the remaining chunk of the angiotensinogen protein. Moreover, angiotensin 2, also colored purple, is cleaved from angiotensin 1 by ACE or angiotensin converting enzyme between phenylalanine 8 and histidine 9. Angiotensinogen is coded by the AGT gene, which is found in the first chromosome, long Q arm, region 4, band 2 to subband 2. What is the function and mechanism of angiotensinogen? Angiotensinogen, along with several vital proteins including renin, angiotensin converting enzyme or ACE, and angiotensin receptors, is part of the renin-angiotensinogen-aldosterone system that regulates the human blood pressure and fluid balance in the blood. At the initial stage, angiotensinogen is released from the liver, usually in the presence of low blood pressure, and renin is also released by the kidneys to the blood at the same time. Subsequently, renin cleaves angiotensinogen between leucine 10 and valine 11 to release the decapeptide protein, angiotensin 1. Angiotensin converting enzyme, which is found primarily in the vascular endothelium of the lungs and kidney, readily converts angiotensin 1 to angiotensin 2. Several alternative pathways also exist, but for the sake of this video, we won't be discussing them. Angiotensin 2 is the major hormone of the RAS that directly affects the arteries, kidney, adrenal cortex, and the brain, which ultimately increase blood pressure through vasoconstriction and increasing blood volume. Let us go further to angiotensin 2. First, angiotensin 2 binds with angiotensin receptors type 1 found in the walls of vascular endothelium, blocking the release of nitric oxide, which is a known vasodilator, hence constriction of the arteries and subsequently raising blood pressure. Second, angiotensin 2 is degraded by peptidases to angiotensin 3, which stimulates aldosterone release in the adrenal cortex. Aldosterone is a steroid hormone that works by increasing sodium reabsorption and potassium excretion. Hence, fluid is pulled to the blood, increasing blood volume, which results to increasing blood pressure. Third, angiotensin 2 also directly affects the kidney by increasing sodium reabsorption, similar to the effects of aldosterone, although much quicker. Lastly, angiotensin 2 interacts with the brain in three ways. Number 1, by binding to the hypothalamus that stimulates thirst to increase fluid volume in the blood. Number 2, by stimulating ADH or antidiuretic hormone or vasopressin in causing the kidneys to decrease urine output and retain water in the blood. And number 3, by diminishing sensitivity of the baroreceptor reflex responsible for counteracting the effects of RAS. The renin-angiotensinogen aldosterone system is regulated by decreasing renin output in response to increased blood volume sensed by stretch receptors. Renin reduction is also regulated by increased levels of angiotensin 2, causing a negative feedback loop. Now, what happens when angiotensinogen is impaired? The renal tubular dysgenesis. It is a severe fetal disorder due to an autosomal recessive disease where mutations happen in the angiotensinogen and other components of RAS, causing poor development of the kidney and other tissues, which can be highly fatal to the baby. Initially, mutations in the angiotensinogen gene and other RAS components like ACE gene, renin gene, and angiotensin type 1 receptor gene causes impaired renin-angiotensinogen aldosterone system, which leads to failure of the kidney to control blood pressure. This malfunction causes hypoperfusion or low blood circulation due to persistent low blood pressure, reducing the oxygen delivered to developing fetal tissues, which leads to several debilitating effects on the fetus, including underdeveloped lungs, which contributes to fatal death due to respiratory failure, and impaired kidney development, which progress to different effects like persistent hypotension or low blood pressure. and anuria, or the reduced to no urine production due to kidney failure. Anuria leads to oligohydramnios, or the reduced amniotic fluid that serves as cushion for the developing baby in the womb. That leads to the characteristic effects of Potter sequence like flat nose, wide low-hanging ears, excess skin, club feet, and ossifications or holes in the scalp. In conclusion, angiotensinogen is an important component in the regulation of blood pressure and fluid homeostasis, process known as the renin-angiotensinogen-aldosterone system. It is also the only known precursor of all angiotensin peptides. To learn more about other proteins and their function, check out our website at bsedsciencepup.com, because learning science is fun. Thank you!